Now in meiosis there are two processes, meiosis-I and meiosis-II. So supposing if this is a parent cell, it divides into two daughter cells. In the parent cell, supposing if it's a human cell, then there are 46 chromosomes in the parent cell. When it divides into two daughter cells, then there are 23 chromosomes. That means it reduces. In meiosis, the number of chromosomes reduces, and each of the daughter cell will be having 23 chromosomes. So the formation of two daughter cell from one parent cell is known as meiosis-I. Now this again divide into two cells and four cells are formed, four daughter cells are formed. Since it takes place by mitosis, this formation of the four daughter cell, and each daughter cell will be having 23 chromosomes. So the forming of four daughter cell from two daughter cell is known as meiosis-II. And they look completely, and all these four daughter cell look completely different from the parent cell. So in meiosis, the number of chromosome gets reduced, and all the four daughter cell does not look same like the parent cell. In mitosis, the number of chromosome does not get reduced, it remains the same, and they look exactly same like the parent cell. Now in meiosis, we have studied, I have discussed before. In meiosis takes place in germ cells, that is the reproductive cells. Now what are the reproductive cells? That is the gametes. Now what are gametes? Gametes are sperm which is found in the men and egg which is found in the woman. Now this sperm has 23 chromosomes and egg also has 23 chromosomes. Both combine to form a single cell zygote. Now this 23 chromosome from sperm and this 23 chromosome from egg combine together to form zygote, and the zygote will be having 46 chromosomes. So the meiosis comes to end over here. Up to the zygote formation, meiosis take place. After the zygote formation, mitosis take place. Now all living organisms, whether it's a human being or the puppies or the kittens, all their life begins from the single cell zygote. This single cell zygote again divides and redivides again and again by mitosis to form different organs like the heart, liver, limbs, etc. And the offsprings which are born, again by mitosis, it divides and the puppies grow into, because of the mitosis division of the somatic cell, the puppies grows into dog and the kitten grows into a cat. So I repeat once more, meiosis occurs in germ cells. meiosis जो है वो germ cell, meiosis जो रहता है उसका दो प्रोसेस है, meiosis-I, meiosis-II. parent cell, समझो ये parent cell है, इसमें 46 ह्यूमन सेल लेते हैं तो ह्यूमन सेल में 46 क्रोमोसोम्स है. तो ये पेरेंट सेल डिवाइड होके दो सेल बनता है, लेकिन इसमें क्रोमोसोम्स आधा आधा होता है, रिड्यूस हो जाता है. इसमें 23 जाता है और इसमें 23 जाता है. ये दो सेल बनता है, उसको बोलता है meiosis-I. और ये डॉटर सेल अगेन डिवाइड होके 4 डॉटर सेल्स बनता है, मगर नंबर ऑफ क्रोमोसोम्स इसमें इसका जितना है उतना ही रहता है, कम नहीं होता है. तो वाई इट डज नॉट रेड्यूस? बिकॉज ऑफ माइटोसिस. इसमें माइटोसिस, मियोसिस जो रहता है माइटोसिस के जैसे ही है. तो 4 डॉटर सेल बनता है, उसको बोलते हैं meiosis-II. अभी मियोसिस किस में होता है? जर्म सेल्स में होता है. जर्म सेल्स का दूसरा नाम क्या है? रिप्रोडक्टिव सेल्स. रिप्रोडक्टिव सेल्स क्या होता है? गैमेट्स. गैमेट्स क्या है? स्पर्म्स और एग्स को गैमेट बोलते हैं। स्पर्म जो रहता है वो मेन में रहता है, एग जो रहता है वो वुमेन में रहता है। स्पर्म में भी 23 क्रोमोसोम्स है, एग में भी 23 क्रोमोसोम्स है। ये दोनों कंबाइन होके फर्टिलाइज होके सिंगल सेल जाइगोट बनता है। तो 23 क्रोमोसोम स्पर्म का और 23 क्रोमोसोम एग का दोनों कंबाइन होके 46 क्रोमोसोम्स बनता है। अभी यहाँ मियोसिस ख़त्म होता है और माइटोसिस स्टार्ट होता है। तो जाइगोट जो रहता है हर लिविंग ऑर्गेनिज्म, भले वो ह्यूमन बीइंग हो, डॉग हो, कैट हो, पपीज़, किटन्स, उनका शुरुआत हमेशा सिंगल सेल से ही होते हैं। ये सिंगल सेल डिवाइड, रीडिवाइड, अगेन एंड अगेन डिवाइड माइटोसिस से होता है और अलग अलग ऑर्गन्स बनता है। और ये ऑफस्प्रिंग जब भी बॉर्न होता है तो वापस माइटोसिस ही होता है उनमें, सोमैटिक सेल्स में माइटोसिस होके पपीज़ जो रहता है वो डॉग, फुल फ्लेजेड डॉग बन जाता है और किटन जो रहता है वो कैट बन जाता है। तो माइटोसिस और मियोसिस का डिफरेंस वही है। माइटोसिस में नंबर ऑफ क्रोमोसोम सेम रहता है मगर मियोसिस में नंबर ऑफ क्रोमोसोम रेड्यूस होता है। माइटोसिस में जो डॉटर सेल्स है वो पेरेंट सेल के जैसे ही दिखता है और मियोसिस में जो डॉटर सेल्स है वो पेरेंट सेल से एकदम अलग दिखता है। Now meiosis is divided into two steps. We had discussed before that meiosis 1 and meiosis 2. In meiosis 1 there are 4 stages, that is prophase 1, metaphase 1, Anaphase One and Telophase One. Same way in meiosis 2 there are 4 stages, prophase 2, metaphase 2, Anaphase 2 and Telophase 2. Always in meiosis it begins with chirokinesis, like mitotic division. And after telophase it ends with cytokinesis. Same way in meiosis 2 also it starts with chirokinesis and after telophase it ends with cytokinesis. Now in meiosis 1, the longest stage in meiosis 1 is prophase 1. It is the most complex stage. It is divided into 5 substages. What are the 5 substages? First one is leptotene. Second one is zygotin. Third one is pachytene. Fourth one is diplotene. And the last one is dichinesis. तो मियोसिस में 2 स्टेप रहता है, मियोसिस 1, मियोसिस 2। मियोसिस 1 में 4 स्टेजेस है, प्रोफेस 1, मेटाफेस 1, एनाफेस 1, टेलोफेस 1। वैसे ही मियोसिस 2 में भी 4 स्टेजेस है, प्रोफेस 2, मेटाफेस 2, एनाफेस 2, टेलोफेस 2। और जैसा माइटोटिक में शुरू होता है कायरोकाइनेसिस से और टेलोफेस के बाद साइटोकाइनेसिस होता है, वैसे मियोसिस 2 में भी शुरू होता है कायरोकाइनेसिस और टेलोफेस 2 के बाद साइटोकाइनेसिस होता है। मियोसिस 1 में सबसे लंबा और कॉम्प्लेक्स स्टेज कौन सा है? प्रोफेस 1। इसमें 5 सब स्टेजेस है। फर्स्ट वाला है लेप्टोटीन, सेकेंड वाला है जाइगोटीन, थर्ड वाला पैचेटीन, फोर्थ वाला डिप्लोटीन और फिफ्थ वाला डाइकाइनेसिस। Now in 10th you don't have to study much about meiosis, but you have to describe this 5 substages of prophase 1. In short, you just understand this much, and this I'll explain in detail because there is one back question based on this. So I will discuss now prophase 1, the 5 substages of prophase 1. Now in meiosis 1, prophase 1, the first step, prophase 1, has 5 substages, and we have a question 5C. 5C question is there, which is based on that. What is 5C? You have to explain the diagram, what are the different stages of the prophase 1. Now before prophase 1, the cell has a nucleus. The nucleus has a nuclear membrane. With nucleolus in the middle, and centrioles are there. These are the centroid, and inside the nucleus we have the DNA material. So we have the DNA material inside. Now the first stage of prophase 1 is known as what? Leptothene. In leptothene, the nucleus remains as it is with the nuclear membrane, and nucleolus is also there. Centrioles are also there as it is, but the chromatids become condenses and becomes short, thin-like structure. So in leptothene, the chromatid starts condensing. Now the next stage is known as zygotin. In zygotin, the nuclear membrane, the nucleus remains as it is with the nuclear membrane, with the nucleolus inside it. The centroid also there as it is. Now the chromosomes tries to arrange themselves side by side. Now supposing if this is the homologous, the chromosomes which is coming from the father, and let us assume the red color has the chromosome which is coming from the mother. These two chromosomes arrange side by side, and these two are known as what? Homologous chromosomes. These are known as homologous chromosomes. Now what is the meaning of homologous chromosomes? Homologous chromosomes. It is also known as sister chromatids. Non-sister, not sister, sister, non-sister chromatids. Non-sister chromatids. Now we had studied in mitosis sister chromatids. Now what is the difference between sister chromatids and the non-sister chromatids? The non-sister chromatid is also known as homologous. Now the sister chromatids, they are all same, they are all of same size, they have same genes, genes are located at same location, and they have same alleles. In case of non-sister chromatids, they are also of same size. They are all of same size. They have same genes. They are also located at same location, but they have different alleles. तो होमोलोगस क्रोमोसोम, एक क्रोमोसोम जो रहता है फादर से आता है और एक क्रोमोसोम रहता है मदर से आता है, दोनों भी अलग अलग है, इसके लिए इसको होमोलोगस क्रोमोसोम बोलता है। होमोलोगस क्रोमोसोम्स का दूसरा नाम क्या है? नॉन सिस्टर क्रोमैटिड्स। माइटोसिस में अपन क्या पढ़ा था? सिस्टर क्रोमैटिड्स। अब इन दोनों में फ़र्क क्या है? सिस्टर क्रोमैटिड्स और नॉन सिस्टर क्रोमैटिड्स में फ़र्क क्या है? सिस्टर क्रोमैटिड्स सब सेम साइज़ का होता है, सब के पास सेम जीन्स रहता है, जीन सेम लोकेशन में रहता है और सेम एलेल्स भी रहता है। नॉन सिस्टर क्रोमैटिड्स या होमोलोगस क्रोमोसोम का डिफरेंस क्या है? वो भी सब सेम साइज़ है, सेम जीन्स है, लोकेशन सबका सेम है, मगर उनके पास डिफरेंट एलील्स है। अब ये डिफरेंट एलील्स का मतलब क्या होता है? सपोजिंग पी प्लांट यू हैड स्टडीड लास्ट ईयर. Last year we had studied about the monohybrid cross and the dihybrid cross. Both are pea plant, but one of the character, let us take one of the characteristic like that of the seed. We had studied that seed can be round or wrinkle. So if the seeds are round and one of the pea plant is having round seeds and the other one is having wrinkle seeds, then we say that they are of different alleles. Even in human beings, if you say all the human beings have eyes, nose, hands, legs, everything is same, but yet they don't look similar to each other. That is because of the different alleles. So that is the difference between homologous chromosome or non-sister chromatids and sister chromatids. So since one of the chromosome is coming from father and the other chromosome is coming from mother, hence they are known as homologous chromosomes. Now next step is what? Pachytene. So next step is pachytene. Pachytene is the important step. Why? Here also the nuclear membrane remains as it is, the nucleolus remain as it is, the centroid also remain as it is, but here the crossing over of the chromosome takes place. So crossing over of the chromosomes takes place. So the chromosomes cross over each other or overlaps over each other. Where they overlap, that point is known as chiasmata. So the important step over here is what? That the chromosomes cross over each other, hence the genetic material, exchange of genetic material takes place. Exchange of genetic material takes place. Genetic variation takes place. And because of the genetic variation, evolution takes place. So in pachytene, what happens? The nuclear membrane, the nucleolus and the centroid remains as it is. No changes take place in this. Only the chromosomes overlap each other or cross each other, or recombination occurs. So they recombine with each other, that is, they overlap each other. Because they cross over each other, exchange of genetic material takes place. Because of exchange of genetic material, genetic variation takes place. Because of genetic variation, evolution takes place. Now what is the meaning of this? Exchange of genetic material take place and genetic variation take place. Because of this, there is evolution. Now supposing father and mother have two offsprings. So they have two offsprings. So though the offsprings will be having some characteristics of both father and mother, but yet they don't look similar to their parents. So the two offspring don't look similar to their parents. At the same time, the two offspring, whether they are two brothers, two sisters, or brother and sister, they also won't be looking similar to each other. This is known as genetic variation. So what is genetic variation? अगर समझो father है और mother है और दो ऑफस्प्रिंग है या बच्चे लोग हैं, तो दोनों बच्चे लोग माँ और मदर और फादर के जैसे नहीं दिखते हैं, एग्जैक्ट कॉपी नहीं रहता है उन फादर और मदर। और दोनों भाई या दोनों ऑफस्प्रिंग, या तो भाई लोग रहेगा या तो दो बहन लोग रहेगा या तो भाई या बहन रहेगा, दोनों भी आपस में भी सेम नहीं दिखता है। ये सेम क्यों नहीं दिखता है? बिकॉज ये क्रॉसिंग ओवर टेक्स प्लेस, रिकॉम्बिनेशन होता है, जेनेटिक वेरिएशन होता है। तो जेनेटिक वेरिएशन की वजह से कोई भी ऑफस्प्रिंग अपना फादर और मदर के जैसे नहीं दिखता और आपस में भी सेम नहीं दिखता है। तो इसको बोलता है जेनेटिक वेरिएशन। और जेनेटिक वेरिएशन की वजह से ही एवोल्यूशन होता है। जैसा कि बहुत करोड़ ईयर्स अगो, 2 बिलियन करोड़ ईयर्स अगो अपन कैसे दिखते थे? एप जैसा, लेकिन आज अपन एप जैसे नहीं दिखते बिकॉज ऑफ जेनेटिक वेरिएशन, एंड बिकॉज ऑफ जेनेटिक वेरिएशन एवोल्यूशन हैव टेकन प्लेस। नाउ द नेक्स्ट स्टेप इज व्हाट? डिप्लोटीन। नाउ इन डिप्लोटीन the nuclear membranes start disappearing, the nucleolus also start disappearing, and the centroid starts moving towards the opposite pole, and they are connected to each other by spindle fiber, and the chromosomes try to repel each other. So the chromosome repels each other. So chromosome repel each other. ये exchange of material, जो genetic material exchange हुआ है उसके बारे में है। So they try to repel each other. Now in dichinesis, what happens? The nuclear membrane and nucleolus disappear completely, and the chromosomes repel each other, but they are attached at one end. So at the tip of the chromosome are still attached to each other, and they repel each other. So here also they repel each other, here they repel each other, but they are not able to repel each other. This is dichinesis. So in dichinesis, the chromosomes repel each other, but they are not able to separate, because they are attached at the tip. So they are attached at the tip, and the centroid starts moving at the opposite poles, which are connected to each other by spindle fibers. So the chromosome in dichinesis, the chromosome repel each other, but still they are not able to separate from each other because at the tip they are still attached to each other. In diplotin, the nuclear membrane and the nucleolus start disappearing, the centroid starts moving in the opposite pole, and they are attached to each other by spindle fibers, and the chromosome starts repelling from each other. So these are the 5 sub-stages of prophase 1. Now next after prophase 1 is metaphase 1. In metaphase 1, the chromosomes arrange themselves in the equatorial plane, but they are still attached at the tips. So they are still attached at the tips, and they are connected to each other by the spindle fiber to each other. Now in Anaphase 1 is very important. Here the two chromosomes get separated, and one chromosome moves at this pole and the other chromosome moves at this pole. So one of the chromosome moves on, since I have taken only two chromosomes over here, so one chromosome moves over here and the other chromosome moves over here. That means half the number of chromosome goes on the opposite pole and the other half chromosome comes down in the other pole. So here the number of chromosomes gets reduced to half. To half number of chromosomes. So anaphase is very important because here the number of chromosomes get reduced and the half number of chromosomes. For example, if there is 46 chromosomes, then 23 will go on towards this pole and the other 23 will come towards this pole. The next phase is what? Telophase 1. In telophase 1, again the nuclear membranes appear. The nucleolus also appears and the spindle fibers will disappear, and half number of chromosome will go in this. Half number of chromosome will go in this nucleus, and the other half will come over here. This way, two nuclei are formed. So this way two nuclei are formed. After this, cytokinesis take place and two cells are formed. So two cells will be formed. Two daughter cells will be formed with the respective nucleus. And after this, meiosis-II take place. Meiosis-II is similar like mitosis. So both the cell will undergo meiosis-II, which is same like mitosis only. The steps are all same. This daughter cell will result in two more daughter cell, and this will also result in two daughter cells. So two haploid daughter cell undergoes meiosis, which is similar like mitosis, and results in four daughter cells, with haploid chromosomes. What is the meaning of haploid? Only one set of chromosomes. So in meiosis-II, two haploid daughter cells divides by meiosis-II, which is similar to mitosis, and results in four daughter cells which are having haploid number of chromosome. This year you won't be having, in 10th you will not be having in detail meiosis and all, but you should know the prophase 1 5 substages, and I have explained metaphase 1 and anaphase 1 and telophase 1. Meiosis-II is similar to mitosis only. Over here there are two cells, therefore four daughter cells are formed by meiosis-II. Now, process of gamete production and the process of spore formation, both this occurs by meiosis. Now we have already studied, have I already explained all this. So in short, what we have studied in meiosis, that during this cell division, crossing over occurs between the homologous chromosomes. So there is a crossing over, the exchange of genetic material take place, and hence there is genetic recombination. Hence all the 4 daughter cells, that is all the 4 daughter cells are different from their parent cell, and also all the 4 daughter cells are different from each other. All the 4 will look different from each other. They will not be similar to each other. तो meiosis में अपन ने क्या पढ़ा कि प्रोसेस ऑफ गैमेट प्रोडक्शन और प्रोसेस ऑफ स्पोर फॉर्मेशन, ये दोनों meiosis से होता है. और ये सेल डिवीजन में क्या होता है, क्रॉसिंग ओवर ऑकर्स होता है, क्रोमोसोम क्रॉस ओवर होता है. इसके वजह से होमोलोगस क्रोमोसोम्स, क्रॉसिंग ओवर ऑफ होमोलोगस क्रोमोसोम होता है, एंड हेंस जेनेटिक रिकॉम्बिनेशन होता है. इसके वजह से 4 डॉटर सेल्स बनता है. मगर ये चारों डॉटर सेल्स पेरेंट सेल से एकदम अलग दिखता है, और ये चारों डॉटर सेल आपस में भी बहुत अलग दिखता है, आपस में ईच अदर. सो meiosis का ये सबसे इम्पॉर्टेंट थिंग है, दैट ऑल द 4 डॉटर सेल डोंट लुक सिमिलर टू ईच अदर, and all the 4 daughter cells also don't look similar to their parent cell, which leads to this is because of genetic variation, and which leads to evolution.